Welcome to this lecture on the structure of a neuron. Imagine your body as a massive, bustling city with billions of citizens who need to communicate instantly. If the brain is the central command center, the neurons are the high-speed fiber optic cables connecting every single neighborhood. Today, we are going to explore the physical anatomy of this incredible cell and see exactly how its shape allows you to think, feel, and move. Now that we have our city analogy in mind, let us look closer at the neuron itself. The neuron is the fundamental structural and functional unit of the nervous system. It is highly specialized for intercellular communication. Just like a cell phone receives a text message, processes it, and sends a reply, a neuron is perfectly designed to receive, process, and transmit electrical signals. Without this specialized structure, a stubbed toe would never send a pain signal to your brain, and your brain could never tell your muscles to pull your foot away. Building on that idea of receiving and transmitting, let us break down the anatomy. A neuron is divided into three main regions. First, we have the dendrites, which are branch-like extensions that act as the primary receiving antennas for incoming chemical signals from other cells. Next is the cell body, or soma. This contains the nucleus and essential organelles, serving as the metabolic hub where incoming signals are integrated and processed. Finally, the axon is a long, slender projection that acts like a biological wire, carrying the electrical impulse away from the soma toward other neurons, muscles, or glands. Understanding these three regions leads us to a crucial feature of the axon. Many axons are wrapped in a fatty, insulating layer called the myelin sheath. This sheath has tiny gaps known as nodes of Ranvier that dramatically speed up signal transmission. This unique structure is absolutely critical because it dictates the speed and efficiency of human thought and reflex. It means the difference between pulling your hand away from a hot stove instantly versus suffering a severe burn. Diseases like multiple sclerosis specifically attack this myelin sheath, causing devastating losses in motor control and sensation. Before we move on to the biophysics, let us see this anatomy with a concrete visual. Here you are looking at a multipolar neuron. At the top, you can see the branching dendrites extending from the central soma, which houses the dark circular nucleus. Notice the axon hillock, the cone-shaped region where the soma tapers into the axon. Moving down the axon, you can clearly see the segmented yellowish myelin sheath wrapping around it, interrupted by the small gaps, which are the nodes of Ranvier. Finally, at the bottom, the axon splits into multiple axon terminals, ready to pass the signal to the next cell. Now that we have visualized the neuron, let us understand how its physical size affects its function. We must look specifically at the physical size of the cell body, or soma. The soma is roughly spherical, and its surface area determines how much cell membrane is available to store electrical charge. This property is known as membrane capacitance. A larger surface area means more space for ion channels and a greater ability to store electrical energy before firing a signal. This mathematical relationship perfectly illustrates why larger neurons behave differently than smaller ones in our nervous system. This concept of size brings us to our first geometric equation. To find the capacitance, we first need the surface area of the spherical soma. The formula is A equals 4 times pi times r squared. In this equation, A represents the total surface area of the neuronal soma, measured in square micrometers. The symbol pi is the mathematical constant, approximately 3.14159. The variable r represents the radius of the spherical cell body, also measured in micrometers. This formula tells us that the surface area grows with the square of its radius. If the radius increases, the surface area expands exponentially, providing vastly more space for electrical components. Now that we have a way to calculate the surface area, we can determine the electrical storage capacity. This biophysical equation is C equals C sub m times A. The variable C stands for the total membrane capacitance of the neuron, measured in picofarads. The symbol C sub m represents the specific membrane capacitance, which is a biological constant for cell membranes, typically about 0.01 picofarads per square micrometer. The variable A is the surface area we just discussed. This formula simply states that the total electrical storage capacity is directly proportional to its surface area. When the surface area increases, the neuron can hold more charge, requiring a stronger stimulus to trigger an impulse. Let us apply these formulas to a concrete example. Imagine we are examining a small interneuron in the human spinal cord with a perfectly spherical cell body and a radius of 5 micrometers. We know the specific membrane capacitance is 0.01 picofarads per square micrometer. We need to calculate its total membrane capacitance. First, we substitute the radius of 5 into our surface area equation. Squaring 5 gives 25, and multiplying by 4 and pi gives a surface area of approximately 314.16 square micrometers. Next, we multiply this area by our biological constant of 0.01. This yields a final membrane capacitance of 3.14 picofarads. Because this storage tank is small, it takes very little electrical current to trigger an action potential, making this neuron highly sensitive. Now that we have seen a small cell, let us look at a much larger one, a lower motor neuron that controls the heavy muscles in your leg. This neuron has a massive radius of 20 micrometers. We want to find its total membrane capacitance to see how its large structure changes its electrical behavior. We use the exact same two-step strategy. Plugging 20 into the surface area formula, squaring it gives 400, resulting in a massive surface area of approximately 5,026.55 square micrometers. We then multiplied this massive area by our constant of 0.01. This gives a final membrane capacitance of 50.27 picofarads. This large motor neuron can hold much more charge, meaning it requires a significantly larger electrical stimulus from the brain to trigger a muscle contraction, preventing random twitching. These two examples we just solved reveal a profound physiological truth. Notice that while the radius of the motor neuron was only 4 times larger than the interneuron, its surface area and total capacitance increased by a factor of 16. This is due to the squared term in our geometric formula. Physically, this means the large motor neuron has a much bigger electrical storage tank. Because it can hold so much more charge, it requires a significantly larger electrical stimulus from the brain to actually trigger a muscle contraction. This structural difference ensures that our heavy leg muscles do not twitch randomly at every tiny accidental signal, providing stability and controlled movement. We have covered a lot of ground today, so let us summarize the most important ideas. First, the structure of a neuron is a masterpiece of biological engineering, perfectly tailored for rapid communication across vast distances in the body. Second, its anatomy is highly specialized. Dendrites receive messages, the soma processes them, and the axon transmits them. Finally, as we saw through the mathematics of membrane capacitance, the physical size and shape of the neuron directly dictate its electrical properties and sensitivity. Understanding this anatomy is the foundational key to unlocking the complex mysteries of the human nervous system. Great job following along with these concepts and calculations.